Now, as I mentioned in the last lecture, there are four main types of large biological molecules, also called bio or macromolecules, that living organisms synthesize: proteins, carbohydrates, nucleic acids, and lipids. This lecture examines proteins. Whether you eat meat, cheese, or tofu, you are consuming the protein that has been made by another living organism, and proteins are extremely important for life. Proteins serve many functions in living organisms. Structural proteins help organize and shape cells, as well as their extracellular environments. Proteins can act like microscopic motors, moving components around in a cell. The proteins actin and myosin work in concert to contract the muscles in your body. Proteins form an important part of the immune system, antibodies, which tag foreign invaders for destruction by white blood cells. Storage proteins are used by developing organisms. The white part of an egg contains a protein called albumin, which is used by the growing chick. Some hormones are composed of proteins. Hormones are molecular signals that start, stop, or prolong a specific event. For example, growth hormones stimulate growth, cell reproduction, and regeneration in humans and other animals. And last, but certainly not least, enzymes. The vast majority of enzymes are proteins. Enzymes speed up or catalyze chemical reactions that would otherwise occur very slowly. And since chemical reactions are vital to life, enzymes are essential in biology. Amino acids are the monomers that make up protein polymers, and amino acids consist of a carbon surrounded by four groups: an amino, a carboxyl, a hydrogen, and a side chain, or R group. The side chain is variable from amino acid to amino acid. Here's the central carbon with an amino functional group, a carboxyl group, a hydrogen, and the side chain or R group. This is the important one, as is the only group that differs in the 20 amino acids found in organisms. If you recall, the amino and carboxyl groups have special properties in neutral pHs. There are three main types of amino acids based on their side chains: nonpolar, polar, and charged. These are the nonpolar amino acids. The side chain of each amino acid is in the colored area. The letter in parentheses by the name and the three-letter code underneath the name are two different ways you can abbreviate the amino acid. The nonpolar amino acids have side chains that do not have a complete or partial electric charge, and are therefore not soluble in water because they do not interact with polar water molecules. On the other hand, the polar amino acids do have partially charged side chains. They are soluble in water because the chains will interact with water. The last group includes electrically charged amino acids that have either acidic or basic side chains. The acidic side chains readily lose hydrogen ions to the surrounding neutral solution and take on a negative charge. The basic side chains readily gain hydrogen ions from the surrounding neutral solution and take on a positive charge. These amino acids are highly soluble in water. This diagram is not one you need to memorize, but shows the range of solubility for the amino acids. Remember that if something is hydrophilic, it will dissolve in water, while hydrophobic substances will not. To form proteins, the amino acids must be connected together. As with any biomolecule, the monomers are bound to each other with a covalent bond by a condensation reaction. However, we give a special name to the bonds found between amino acids: peptide bonds. These bonds occur between the amino functional group of one amino acid and the carboxyl functional group of another amino acid. Now let's look at the formation of a peptide bond between two ionized amino acids in a cell. Here's the carboxyl of glutamate and the amino of cysteine. When a bond is formed, we have to remove a water molecule, as this is a condensation reaction. This results in the formation of the covalent bond, specifically a peptide bond, between the carboxyl end of the glutamate and the amino end of the cysteine. We call a short string of amino acids a peptide, but as more amino acids are added, the result is a polypeptide chain. We call this growing chain of amino acids a polypeptide because it is not complete. A few rules concern chains of amino acids. First, one end of the chain is called the N-terminus because the amino functional group is found on that side. The other end is called the C-terminus because a carboxyl group is on that side. And we number the amino acids from the N-terminus. In reality, most proteins consist of hundreds or thousands of amino acids, and these long chains can form very complex shapes. We can look at 4 different levels of protein structure: primary, secondary, tertiary, and quaternary. Let's look at primary first. It is just the sequence of amino acids. For example, in this chain we have the sequence glycine, proline, threonine, glycine, threonine, and so forth. If you compare 2 different proteins, they have a different sequence of amino acids and most likely have a different number of amino acids. Proteins are also organized into secondary structures called alpha helices or beta-pleated sheets. Hydrogen bonds between atoms connected to the backbone hold these structures together. These bonds occur between groups that are not close together in the primary sequence. The bonds also do not occur between atoms in the R groups. The hydrogen bonds that form in an alpha helix twist the polypeptide into a spiral shape, while the hydrogen bonds in a beta-pleated sheet fold the polypeptide into an accordion or folded fan shape. Tertiary structure is very complex and results in the 3-dimensional shape of the polypeptide chain. As you can see here, we have some beta-pleated sheets in the secondary structure. The hydrogen bonds are holding the chain in this folded shape, but the side chains can be attracted to or repelled by each other, and this results in a 3-dimensional shape. The shape of the protein will determine what it does in an organism, so the tertiary structure is very important. The different interactions between the functional groups include hydrogen bonds other than those at the secondary level, disulfide bonds, ionic bonds, and hydrophobic interactions. In this tertiary structure, we can find hydrogen bonds between a carbonyl group of an amino acid and the side chain of another amino acid, a hydrogen bond between 2 side chains, some hydrophobic or nonpolar side chains that are clustering together as they are excluded from the surrounding water, a disulfide bond between 2 cysteine side chains, and an ionic bond between 2 side chains that have ionized. All these interactions fold the polypeptide chain into an intricate 3-dimensional shape. If the protein only consists of one chain, we can call the structure a protein at this state. But many proteins consist of more than one polypeptide chain, so they have a quaternary structure. Hemoglobin, the molecular complex that carries oxygen in red blood cells, has a quaternary structure. It consists of 4 polypeptide chains, 2 alpha chains, and 2 beta chains. This forms the perfect shape to hold iron, which is what oxygen binds to. Let me emphasize, the shape of the protein determines its function. Proteins can be many different shapes. The TATA box binding protein is the right shape to bind to DNA. The porin protein is the right shape to form a tube and allow substances into the cell. But what happens when a protein is the wrong shape? Let's look at an example. Remember we discussed hemoglobin? Well, these are normal cells because they have normal hemoglobin. The amino acid in the 6th position is glutamate. But what if we were to change that one amino acid? We would be changing the primary structure, and this might affect the other levels of organization. In fact, a mutation that results in valine at the 6th position creates hemoglobin that is not normal. With this one amino acid change, the hemoglobin forms a crystal structure and is not the best shape for iron, so oxygen does not bind as well. In addition, the abnormal hemoglobin causes the cells to form a sickle shape, and they tend to clump together in small blood vessels. People with this condition have sickle cell anemia and experience many ailments, such as low oxygen levels, aneurysms, and organ failure. all from one amino acid change. Proteins also depend upon a certain environment. When the environment is not right, too acidic, or the wrong temperature, the secondary and tertiary interactions might fail and the protein will unfold. This is known as protein denaturation. When the hydrogen and disulfide bonds were broken in this ribonuclease protein, it denatured and could not perform its job. Fortunately, the bonds can be reformed and the protein reactivated in most circumstances in the body. Sadly, this is not true for extremely high temperatures. The white of the egg contains albumin protein, and when cooked, becomes completely denatured, and Mr. Humpty Dumpty's bonds cannot be reformed.